Hello everyone, today I will be discussing about my research project that discusses the proteomics studies performed on NLRP3 inflammasome activated immune cells. I am Aurchie Rahman and my colleagues Dr. Abuhena Mustafa Kamal and Dr. Joyantuke Chakraborty are members of the bioanalytical laboratory of Dr. Saiful M. Chaudhary at the Department of Chemistry and Biochemistry. So my research discusses about the inflammasome complex NLRP3. This complex is formed whenever there is a danger signal to the cells. By danger signal, we mean whenever there is a cellular stress caused by some changes in the metabolism, or there is some microbial attack to the cells are in stress. In the stress situations, the inactive form of NLRP3 proteins are highly expressed, and with the help of NEK protein and with the help of signals from ATP molecules, these inactive NLRP3s undergo conformational changes and they are formed into active NLRP3 subunits. And these NLRP3s, which are activated now, gather together and they form the NLRP3 inflammasome complex. These complexes then participate in further chemical reactions in the pathways, and ultimately the cytokines IL-1 beta and IL-18 are produced. In this experiment, we chose the human monocyte cell lines THP1 for our experiments. So 4 different sets of cells were prepared. The first was control, the second was with LPS treatment, the third was with MSU treatment, and the fourth was what? With LPS and MSU treatment. So LPS is the lipopolysaccharide, which is an abundant molecule of the bacterial cell walls, and MSUs are monosodium urate crystals, which can activate NLRP3 inflammasomes. So with these 4 different cells, we did the cell lysis, the protein purification, the in solution digestion, and then we did the LC-MS analysis. After the analysis, we did some database search which identified some important PSMs. PSMs are peptide spectral matches. These are the peptide spectra that are specific to different proteins. So by identifying PSMs, we are able to identify the different proteins that are expressed in different set of treatments. After identifying the PSMs, we did the quantitative analysis with them, and then we did some bioinformatic analysis on them. So in our research, we saw that there is around 1500 different proteins expressed in the control and BBS set, and around 750 proteins expressed in the LPS, in the MSU, and in the LPS MSU treatment sets of the cells. A Venn diagram was constructed using the data we got, and the diagram showed us that there are some common proteins that are expressed in different set of treatments, and there are some exclusive proteins that are expressed in specific treatments of the cells. A construction of the heat map showed us the differential expression pattern of different proteins of these treatments for the cells. Then we performed the gene ontology analysis of the identified proteins, and we saw their cellular components, we saw the biological processes they were participating in, and we saw the molecular functions they were performing. After all this analysis, we were able to identify 6 different proteins that are exclusively identified in the NLRP3 activated macrophage cells, and we were able to see that the identified proteins were mainly participating in the catalytic and binding activity of the cells. So we are hopeful that in future, with the affinity purification cross-linking mass spectrometry, we will be able to identify some novel communicators of this NLRP3 interactome pathways. Thank you all for your patient hearing.